Good afternoon, everyone. Today we are going to have an interesting topic on 3D bioprinting. So we have three speakers here today. The basics will be delivered by Dr. Shibu. He is from IIT, who has been working in 3D printing, organoid development, and tissue patch, organ development, etc. And then followed by what we have done in our laboratory on the esophageal regeneration. Dr. Pavan will talk about how we have developed an esophageal composite. And then Dr. Rohit Dama will talk about how we have tested this esophageal composite in animals, mainly the rabbits. I invite Dr. Shibu to take.
Thanks, Chikela, inviting me for presenting my work. The heading, actually, I have written transforming. I'm going to use some other conventional word translation. The reason is still we need to make a lot of changes in current system where currently we are not, maybe I am from academics for many, and there is hardly like some interactions happens between the academic peoples and clinicians. So we used to travel from IIT to city to meet one clinician. So I'm very happy to see. Maybe I think many clinicians must be listening this. So I'm very happy for that. So that's the reason. Like we need a transformation to develop something. So that's why purposefully I kept the word transforming, not the word translation. So the current trend, the recent developments in India is really supportive for medical devices, and it is almost 1 crore like sector in India, and but we are contributing only 1.5% of the global share. That is the very something sad that we are not able to raise from there. And maybe I'm not going through each and every line. The basic idea is like if you go through the recent medical device sector policy, there the last 2 points what I have mentioned here, implantable biologics and tissue models are, it's not written there because it's not there in India. There is no implantable biologics. Maybe one of like a few biologics that we are importing from abroad and we are using. So, but remember there is a raising demand for such kind of implantables, maybe that clinicians like you know better than me. So what are these implantable biologics? These are actually the products of the research area called tissue engineering. So the field of tissue engineering has been trying to develop tissue substitutes for last 20 years or more than 20. Maybe since 1980s, we have been trying to develop something that can patient. So that is actually an interaction or maybe the merging of two branches like medical field and the engineering or the biology. So that is called tissue engineering. So what tissue engineering promise? It promise extracellular matrix and viable cells in an extracellular matrix and the cell function. Means if you are developing something in lab in vitro conditions, these things are possible, like biological complexities can be mimicked, cell function can be mimicked, and the cells will be viable, and there will be an extracellular matrix. So now what happens, like recently, maybe in 2 decades, there is a major development in this that is called 3D bioprinting. So we can mimic tissue and organ architecture. So before, if you see the conventional technology here, these are actually mold prepared structures. There we'll take a negative mold and we prepare some scaffold. There we will put the cells, and then that is how we used to prepare tissues. And now bioprinting is an emerging field which can completely mimic the architecture of an organ or tissue or whatever you want. So how is it possible? Like suppose you have a design, a digital design. It can be MRI, CT, whatever it is. You can extract the images from there, and you can develop, like, it's a digital design, and then that you just need to give to the printer. It can print as per the design. So this is actually the principles of 3D bioprinting, means the basics of 3D bioprinting. So, but how far, like, where we are now, that is actually a question. Now, like, suppose if there is a machine that can make potato chips, you just need to give potatoes. Maybe what's my understanding is, then you will get the output like chips. There you need to feed everything, you will get whatever you want, like in a highly reproducible and precision. Okay, so there is no, you cannot find much chips which is statistically variant from other chips. So that is actually what you call, in 3D bioprinting also it is possible, but instead of potatoes we will be giving biomaterials. So these biomaterials actually, it is not that easy to handle. Okay. So that I'll come to that. So now, how this 3D bioprinting works, the very basics, it is actually additive manufacturing. Means the instrument put layers over layer, and then one by one it laid some biomaterials along with cells, and thereby it can get a complete architecture. So here if you see, this is the CAD design that I have prepared from a cornea. The machine cannot print it as such. So what the machine does first, it takes and then it slices the image. It gives multiple slices of the organ or tissue, whatever you want. Then the print head, like you can see here, the nose is actually the accessories that just for the convenience I have kept it there. So this nozzle can move as per the layers, whatever the layer is given or the machine has sliced. So based on that, the nozzle moves and deposit the ink. This is how the 3D bioprinting works. And right hand side you can see this is something called bio ink. Instead of potatoes in the like a food processing unit, have a bio ink here, but this is highly variable. It's not something like a potato. It's highly variable and very difficult to handle. And why, that I can tell you later. So then there is a syringe that is attached to the instrument. So there we will fill this hydrogel, and then along with the cells or other material factors, then it will extrude through the nozzle. So now based on this design, the biomaterial will get extruded through the nozzle, and it will deposit layer by layer till it reaches complete structure. So this is how the bioprinter works. Now, this bio ink. Okay, this bio ink has many things, like it has polymer, because we cannot just print cells. Because if you print cells, okay, cells will fall somewhere, that's it. But it cannot grow, or it cannot, it has some space. Here, first what we do, we take appropriate biomaterials, that most probably it will be polymer, a protein, it can be any biological substances which has some particular viscosity. It should have some particular viscosity, only it can go through the nozzle. And in there also, like in that viscosity also, there are some properties that maybe we cannot discuss now. Then we mix the cells, whatever the cells of interest, that we mix with the biomaterials, and then load into the syringe, and then we'll give the command based on the digital design that we have. So it will get printed. Now, there are three types of printers. Maybe most of you know the extrusion-based bioprinting, where cells and materials gets extruded. But here we have many other printers also, like inkjet-based printing, the very first printer that has been developed from Epson inkjet, like a printer, our computer printer, that they have modified and made the inkjet bioprinting technique, and also laser-assisted bioprinting. There, that also works based on HP or computer printer. So there we'll be having a biomaterial, and we'll be having a laser, and then laser will go and then cross-link the material as per the design, and then finally we will get a shape here. So this is the most commonly used material in sorry, bioprinter in the medicine or research. That is the extrusion based bioprinting. There we put the hydrogel and then we will get the uh, desired structure. So its application definitely for engineering applications, implantations, and then drug screening, then in vitro screening models. So what is the current status of this 3D bioprinting? I would say this is now if you go any lab, any Indian academics laboratories, they want to get one bioprinter. So it is something like a sexy word now. So everybody is getting, so I also want, but and people does not know that it is not an instrument where you just turn the button and you, there is like if you want to print something which takes 2 hours, you have to sit in front of the bioprinter, see and control. It's just like a driving, so you need to do and make corrections each and every moment. So this is the current situation of bioprinting. But I will never say if you see this image, I want to print a grid with equal whole size in between, but it ended up in this structure. Okay, this is the reason, like it's extrusion and many things there, many hundred parameters are there to be controlled, and also the shape retain or the shape fidelity. Here if you see, I can print maybe one layer here, but after that if I want to print multiple layers on that, then I need such a biomaterial, an ideal biomaterial which can have the mechanical property and also the like shape fidelity and cell supporting or the biocompatibility, biodegradability, and ultimate like the bioprintability. So if you don't have, if you're missing one of these, then you cannot use that particular material for 3D bioprinting. So, but doesn't mean that 3D bioprinter does not have any potential, but it has huge potential because of its reproducibility, high throughput, and also customization. Like we can do any kind of customization. So that's why it is very much applicable for 3D human tissue models. So where now like there's a huge mismatch. Maybe here you cannot find much mismatch. It is exactly matching, but there is a huge mismatch between the efficacy result that comes out after an animal study and the actual clinical outcome. So this is because of the difference in the genetics of animals and human beings. So here actually we can apply this technique very much. Like this, if you are printing one spheroid or one drop of something, it will reproducible like maybe 100 times or 1000 times, and also you need one drop of material. That's why it is called a high throughput technique. So because of these abilities, we can have much like very fascinating applications with the 3D bioprinting. And also when you, if you want to go for customized patient implants, that is also possible in future, but it's not currently it is not in practice, but definitely it has a future. And also now we are working on some like with one company, we are trying to print handheld bioprinters so that clinicians can use it in the OT just by pressing some some buttons. It can extrude the hydrogel so that you can use it. So the discussion is on, and then we are working for that. So now biomaterials, there are multiple types of biomaterials: synthetic, natural, and composite biomaterials, and everything has its own advantages and disadvantages. And the tissue engineering products, which is like now currently available in foreign countries, so autologous biomaterials from autologous tissues, allogenic, and then xeno, synthetic, composite, everything has its own advantages and disadvantages. But here what I am thinking, how can we reach the clinic very fast? So I would like to focus my research in autologous, allogenic, and xenogenic materials. And one more thing, there is no xenogenic products in India. Now only one product got approval from CDSCO. So now a little bit about me. Like I'm here to what to say. I want to propose something to AIG. So I want to develop like a lab which completely focus on cutting edge research, and also the main focus would be innovative biomaterials, injectable implants, tissue substitutes, and preclinical human models. So because there is such like if you whenever I talk to any clinicians, they are saying bring come up with something. So I think in such a environment here, like in AIG, what you have, it is very much possible. And this is about like my credentials. I have been in this field for last 12 years and filed some patents, published papers, and fortunately, two products are in clinic, like it's under clinical trial now at LV Prasad Institute. That is mainly cornea for two diseases. One is SCAR and also other one is for keratoconus. These are under clinical trials now. Yeah. So this is a short story behind the restoring vision. That was actually my PhD topic. And here we like what did I do exactly? Like there is already some technique for removing cells from the tissue, because otherwise you cannot take many of these tissues into clinic because of the like immune rejection. So what did I do? Like I have used only sodium chloride to remove the cells from the extracellular matrix. So I have collected cornea, discarded cornea from eye banks and also from slaughterhouses, some meat export companies, and then collected the cornea, removed the cells by using sodium chloride, and then quantified it, characterized it for extracellular matrix content, DNA content, and then rheological parameters, many things we have characterized. And then the basics of this hydrogel is, this is all like a collagen. So what we do, we take the collagen, we'll remove the cells, and we will unwind it. Like we'll remove the collagen fibrils, and then by applying temperature or appropriate pH, we will recoil them, so that once you put in the body, it will become a gel. So this is the basic principle of this type of extracellular matrix hydrogel, or like a collagen based hydrogels. And the interesting fact, like it is actually serendipitously I found that these materials actually stopping scar formation. Even if you are putting the scar forming cells in this hydrogel, it will not form, it will not grow as a scar forming cells. It will behave like a normal cells. This is the most striking feature of this hydrogel, or maybe this type of any tissue origin hydrogel. I believe so, but this particular test I have conducted only in corneal matrix hydrogel. Now, these are some small experiments that we have conducted in animal studies. So we have prepared a scar, we made an injury, and there we have filled the hydrogel to check whether ideally it should develop towards scar. Scar should be developed, but what happened here? The scar was not developed, and so here you can see the histological images. Here, the human bovine corneal hydrogel, and this is the human corneal hydrogel applied cases. So both got regenerated, and you can see the extracellular matrix arrangement also. It is actually completely, what is that, recovered or regenerated. And in another experiment, we have created a scar and we kept it for a few months, and then we have surgically removed the scar and then filled the hydrogel. In that case also, we could see some improvement. Like we could see the cornea is getting its thickness back, and also the reepithelialization is happening, and the arrangement of fiber is also almost like normal. So these are very promising result, and it can be extended towards other disease conditions and also other extracellular matrices. So for these, we have received CDSU approval for test manufacturing, and we are going to manufacture in the GMP condition, and we'll be going for preclinical and clinical soon. And this is a cornea that I have printed during my PhD time. So we made a CAD model, the one that I was showing initially, one design. So then the cornea, actually the advantage is cornea does not have any vasculature, but the disadvantage is this cornea have like curvature. It is curved. So printing such kind of structures are very difficult by using extra bioprinters, especially if it is a extracellular matrix hydrogel, which is very delicate and does not have any much mechanical property. So we prepared a mold by using stereolithography. It's another printing technique, and then we have printed the corneal extracellular matrix on top of this, and we could achieve this type of like a curved cornea with curvature. It is almost like a human cornea. This is the comparison. This is the human cornea, and this is the bioprinted cornea. And we have checked its implantability also. We also have conducted animal studies, and perfect, and reepithelialization happened within 8 days. And now the study is on, like it's ongoing, and also we have applied for some bigger grant for continuing this work, and also for clinical trials. So this is the esophageal hydrogel that we have prepared from goat esophagus. We collected the goat esophagus, we have removed the cells from the esophagus, and then prepared hydrogel. And then that we have characterized it for cell viability, rheological properties, many list of characterizations are there. We have conducted that, and then later we bioprinted different structures. So here the idea is to demonstrate that, or to establish that these type of different intricate structures printing is also possible with bioprinting. So here a small ring that we could print, and also a small patch of hydrogel. So here you can see some of the images are in some, the pink color. So this is actually photo cross linking. Like there we have added some chemicals, so that it will start releasing the free radicals, and these free radicals will takes part in the gelation, so that it will be faster. Generally, we don't need to do anything. If you put it in the body, because of the temperature 37, it will gelate. But here, to enhance that, or to speed up that process, we have added cross-linkers. That's why some of these are in pink colors, and we could print these structures. And behind all these things, there's a CAD model that we prepared, and based on that CAD model, we could print this type of multiple structures by using 3D bioprinting. And this is a thickness of 0.5 centimeter and 2.7 centimeter. And here actually, so here if you see, this is actually this ear lobe. But for that particular one, we had to use a scaffold, because this is not self-standing structure. These type of structures are not self-standing. It has some structures that is actually hanging if you place it. So that's why we had to use these type of structures as a scaffold, and then printed on this, and then we could remove it after the gelation. So here pretty like good structures we could achieve. So this type of reproducibility is possible with 3D bioprinting. This is something very interesting. Like we could prepare a patch of esophagus tissue. Like here we have put cells, and we have cross-linked it with eosin Y. That's a chemical that we have used here. So that is actually foldable, unfoldable, and you can do whatever you want. So these are the structures, and it's almost a 7 into 7 centimeter size, and is completely made up of esophagus. And if you compare the chemistry, or the the molecular aspect of like the entire protein composition, everything is more or less similar to human esophagus. So that's why this type of tissue materials, whether it's from xeno source or from human sources, this may play a major role in translating this type of technologies to clinic, because it won't take much time. It does not have much regulatory complications. So so I'm going to show the future plans. So this slide actually showing the limitation of the 3D bioprinting. Here, this type of tubular structures with extracellular matrix is very extremely difficult. So what did we do? We prepared a mold, and by using 3D printing, and then we have filled this esophageal hydrogel into this, and then cultured for few days, maybe 14 days, and then finally we have removed the mold, and we got this type of tubular structures. So mimicking this type of tubular structures are extremely difficult today by using extracellular matrix, because of its poor mechanical property. So now, why India is lagging behind in utilizing these type of technologies? And even if you want something from skin substitutes, we are importing it from abroad, and which is very costly, and mainly because of lack of awareness, and also the high cost that is involved in importing, and then surgical cost, everything, and then the fear of regulatory requirements. People are thinking that regulatory bodies are there to restrict us, but of course they are restricting us, but that is for a better outcome. So, but if you approach them, they are very friendly, so we can go ahead with that. And also cutting edge medical like what to say, medical innovations are not happening in India because of the funding problems, and some this type of interactions are not there, clinicians or researchers. So if we can make that, then definitely that's going to make some big change in India's medical device sector. So here I'm coming up with a new perspective where I would like to completely utilize the waste tissues or whatever you have here, even including the cancer tissues if you have. I would like to utilize these type of tissues for my research here in like AIG, and also that may not serve the purpose. So we also need xenogenic products, that is many waste materials that are throwing out from the slaughterhouses or export companies, meat export companies, that actually a good source of tissue engineered products. So we can utilize all these things for a for clinical applications. And I have the know-how for these many things. I can bioprint materials, and also these tissues, actually I prepared hydrogels and then characterized it for many things, but everything got stuck there in TRL4. Because that, I need the help of clinicians or the such kind of research setups, GMP facilities, and many such facilities are required. And also tissue models can be prepared that can have very good outcome. Suppose if we can screen the drugs for particular patients, so we can use we can test the efficacy before giving to the patients. So these are very much possible with these type of techniques. And so this is a roadmap for this center. First I would like to if I get a chance, I would like to interact with the clinicians, and then we'll be dividing the plan into three, like some small research, and also the medium risk research, and then the pie in the sky, like something high risk and high reward. I don't know whether it's going to work or not, but that involves a very high cost. But if it works, then it will be a big change in this field. So I would like to divide the projects in this type of like multiple categories. And also finally we'll be developing the prototypes, and we'll be going for clinical trials. And once we reach the clinical trials, it can be tested in patients here at AIG. So this is the like a detailed plan where we can explore what to say, some small experiments initially, even small experiments, and then based on that we can go to the medium and then the large scale or big projects that actually going to make some changes. So this is the plan. And it includes the spheroid, organoids, microfluidic devices for drug testing, and also lung, liver, pancreas models. And the last one includes actually many like whether we can reverse fibrosis, or we can stop fibrosis, or we can regenerate the area where it's already like fibrotic. And tissue, like altered tissue can be replaced with normal extracellular matrix. This is called something, a new term that I would like to coin, that is a microenvironment therapy. So this is what exactly I would like to do here. The immediate targets, like first we can target the patients with HCC, where we can go for drug screening. And the next one is the liver cancer microenvironment replacement, where we can replace the, suppose a small area of liver is diseased. I don't know the practicality, that maybe clinicians has to decide, but I have some idea, so I would like to discuss with them and then execute it if possible. So so that area can be removed, and we can fill with a normal hydrogel or normal liver hydrogel, so that the area will become cellularized and then start functioning. And then esophageal and mucosal patches. Then the most interesting thing is the immunotherapy drug delivery. It can be via a catheter. We can deliver the microbeads that is prepared from the biomaterials, and we can deliver the cancer drugs locally at that particular area. And of course there is something we need to work behind, like that is the behind the screen, that the strategies for expediting this type of clinical, we need to identify the problem and why it is not happening. Then we can make this type of wonderful products for the patient, sake of patients. The future plan, though my focus would be on GI or but enterology initially, like to expand it to other tissues and other area of the body tissues and organs. And so this is the mission. Like we'll be starting with some small experiment with what to say, with some seed grant, and we'll be going to the step by step, early stage, late stage validation. And once it is the once the treatment, once the CDSC approval we get, then we can go for the treatment at AIG in a research under research plans, and then we can commercialize it if required, or maybe we can keep it the patents open so that it will be available for everybody. So thank you. So please ask if you have any. That is only muscle. That yes. So that we have seeded with the stem cells. And then if you seed stem cells in this, it will get differentiated into, it will start expressing alpha SMA and all muscle related proteins without any growth factors. Yes.
And the tubal structures you have shown,
Yeah.
they can be customized.
That can be.
Size of
Yes. It can be customized. Yeah. By using 3D printing only, we have prepared the mold for that. 3D, not 3D bioprinting, by 3D printing technology. We made a CAD model, whatever the size we want, we can prepare, and then that has to be printed by using resins, and then that we can use preparing the tubular structure. No, it will be like a tubular structure, but its mechanical property for a in vivo application that has to be tested, but it will stay as such, like it will be in the tubular form. Yes, it will not collapse. But by my cornea that I have printed, that collapse in the sense, if it is in water, it will it will show the curvature, but once you take it, it will be like a drop of water. But once you place it over the eye, then again it will get its structure back. So that's why now we are trying to improve the mechanical property of that.
Excellent talk, Shubhu. My question is how to manage the cell ability printing to conditions. It's a three-layer structure. Manage the cell viability, at the same time how to manage the mechanical properties of the user when we are printing them.
Yes. Yeah. It's a very good question, because this is exactly the challenge that we are facing during printing, but it's met like we can do that. First of all, the mechanical property, that's why I was showing the cross-linking. We have some cross-linkers, photo cross-linkers, by using different wavelength of light, either UV or visible. So we can mix some chemicals, and immediately after printing, we can expose light so that it will stay, it will be stable. And viability during printing, definitely the cell will be going through the nozzle, which is of maybe 50 or 100 micrometer. So they will be viable. There is no issues, because the cell will never experience that much shear force, because it is in a viscous solution. The cells will not get harmed, but at the same time you cannot go less than 50 micrometer, because then it will be very much detrimental to the cells. Cells may die.
Another simple question.
Yeah.
For example, if we are making the esophageal or culture sheet and everything using 3 layers, once we wanted to transplant the one for some other animal model, right? How will you get entered into the long-term period before vascularity is developed? How will it managing?
Yes. Actually, this tubular structure we haven't tried in vitro, first of all, but what we could observe that once you put it in the media, we cultured them for 14 days. The media is able to enter through diffusion because it's a hydrogel. It inherently it has a capacity to absorb water. So it can absorb water and it can reach the innermost layer also. Suppose if it is spheroid or something, and it's a package of cells, so there is no space in between. So if it is spheroid, maybe if you go inside, like after 200 micrometer, it is very difficult to reach the nutrients. But in a this type of hydrogel structure, it is very much possible because the nutrient passage is happening through diffusion. So it can take the water very much inside also. The nutrients will not be a problem. Thank you.
Good afternoon, one and all. I am Dr. Pawan, involved in translation. Before coming into the building blocks of the esophagus and everything, should know, we all know that that advantage and applications of stem cells and biomaterials in related to the translation research. As of now, we all know that using stem cells in translation research attracts more attention in the field of bioengineering. So, achieves stem cells and translation research achieves tissue engineering and organ repair and replacement, wound healing, neuronal disorders, diabetes, and other medical advancements simultaneously. The biomaterial research attracts more attention in the field of regenerative medicine, scaffold-based engineering, during drug delivery, and biocompatible implants and neuronal disorders, artificial organs, and bioengineering. But the synergy between the biomaterials, stem cells, achieves a remarkable achievement. Heard about the remarkable achievements, the synergy and the biomaterial. But sounds like to hear remarkable achievements. But we know that each of the biomaterial has so many limitations after long period of culture or long period of translations also. So the thing is, especially in the conditions of esophageal regeneration, we all know that esophageal is made of three-layer culture, especially epithelial layer and circular muscle and longitudinal. For example, if we wanted to construct the three-layer systems, it's very difficult because of the reason the esophageal construction is much lesser in the 3D printing or as a translation. So the main question is how to overcome the limitations of three-layer culture is a big issue. So for challenging this, there are so many biomaterials are present using and stem cells, and there are so many stem cells are they're synchronizing the cultures also present in that. So to address the biomaterials and the stem cells, so there are so many disorders like esophageal disorders like GRD and Barrett syndrome and esophageal cancers. So we know that the stem cells and the biomaterials are present, people are using for construction of vehicles, but these also having in the long term is also having so many challenges and concerns. For example, foreign body rejection. For example, if we are using allogenic stem cells or some XYZ cells, so by using biomaterial or 3D printing culture, there is a long-term culture conditions. Some, for example, in the pluripotent stem cells, we don't know the cells are in the particular type of cells are intact or else they are converted into some other type of widow. So to addressing this, the foreign body rejection may happen. And another one is injectable injection risk. How to inject the cells in the particular site, how to inject the cells in the particular way of the pattern, that is a big issue. And another one is the mechanical limitations. So as I ask you that Shubhu, how to the mechanical strengths, for example, esophageal is much required in mechanical strengths, each and everything. So how to improve that mechanical strength is again the big challenge. And degradation issues. For example, if we are using some other gelatin, for example, you said the gelatin. So in the most of the literature says the gelatin, once we implant the gelatin, is from emboli. So how to answer that emboli and vascular development, that is a big challenge. Then difficulty with integration. So the material what we are using in the long period, how it integrate into the particular tissue type, that is a big challenge. Then another one is a long-term durability. How much our biomaterial, 3D printed biomaterial are durable, how much it intact into the tissue type and cell type is a big issue. Then surgical complications, these are the main consideration and challenges during the bioprinting and the esophageal regeneration to address these disorders. So how to overcome these challenges is a big issue, right? To overcome these limitations in earlier, so we started to experiment on this SAS material. We started an experiment in related to the one single wrapping system by improvising esophageal regeneration and so on. So initially we brought from the SAS material from the Kurk Biotech, and material efficacy was tested by a biocompatibility assay in vitro and as well as in vivo assays. We did a plan initially. The first experiment we done with the SAS material with integration with the MSS. So the picture here, the showed, these are the cells with SAS material. So here we will observe that we check the different time period patterns. So here we are the cells are very limited, cells are intact. And then again we can check this one. So for example, we can check 12 hours, 24 hours, and 48 hours, 72 hours. So there is no much intact in this. Means that the cells are not adhered. The material is not supporting here. Then we can confirm it again by trypan blue staining. So there are much clumped of trypan blue staining the tapping, and trypan blue stains are present. It's trypan blue positive. It means that the cells are dead. Then again we can confirm by DAPI staining. So there are much intact or else clumped DAPI is present in it. So it means that earlier our observation shows that the biomaterial is not initiating the cell proliferation. Then we can go back and recheck again into the biomaterial is a plain biomaterial in the microscopic observation. We observed that material is with oil droplets. So it means that there is not completely decellularized. And again, the decellularization process or else material process, we can check it by oil red host, whether these cells are which type of cells, or else the oil droplet is there is the oil droplet, some other artifacts we just wanted to check once. So we did oil red host. So these are the oil droplets present in it. And again, the plain nucleated membrane, we can check it is the DAPI staining, the completely nucleated one. So it means that there are nucleases or nuclear materials are present in it. It means that the material is the SAS material what we have brought from this, not completely decellularized. Because of the reason it was not intact in the tissue cells. So again, the question is how to improvise the SAS material for enhancing our experimental suitability. For example, esophageal wrapping. So to addressing this for ASG and AHF department, we developed a SAS material for suitability for esophageal regenerations. So this is pre-treatment. Again, parameters and the post-treated oil drop, and then we can confirm by the oil red was staining, and there is no nuclear in present. It means that there is no nuclear material. So again we initiated, go back to the experiment again we initiated the experiment again with the integration with the SAS material with the MSCs. Here we observed that these 0 hours picture, once we seeded the cell, here SAS material with the 4 hours, the cells are intact. Here we clearly observe. After 7 days, the cells are getting proliferation. And 14 days, the monolayer was found. It means that the material is biocompatible, and now it's encouraging the cell proliferation. Then the live imaging system, the live imaging microscopic image are there in the SAS material. Then the cells are interacted in the material we confound by the calcium with pH staining. Here the green indicates calcium cell present. So based on the in vivo study, so we confirmed that the material in vitro conditioned material is suitable. So it's biocompatible. In vitro biocompatible material is prepared by ISG. Then again we can check this material each and everything by in vivo experiments. So initially we did it through two different ways. Once the material in vivo composite or else the material is in vivo conditions, how the material is suitable, and again how to overcome the dead populated cells by using composite after transplantation. That is a big issue. So to addressing to these issues, so we prepare composite. Once is the SAS PRP thrombin and SAS PRP with the cells and SAS PRP with thrombin cells. So here we done the two experiments. One is a subcutaneously we done the implantation with the composite, and another one is a peritoneal. The subcutaneous initially we implanted that after 4 weeks of the retrieval, we observed that are present but very limited cells. I hope very negligible vascularity is developed in subcutaneous. To understand this, again we confirm. So this material is biocompatible within the in vivo conditions or not. We don't have the addresses on this. Then again, we implanted the same composite into the peritoneum. Here we have observed a remarkable. Yeah. And again, with the cells, composite with the cells, SAS, PRP, thrombin with the cells, developed vascularity, and some of the epithelial cells is in it. And then the vascularity we confirmed by the immunostaining. So the peritoneal composite implantation promotes the vascular development. Here we observed that. So it means that the material is biocompatible and it encouraging the vascularity, and it encouraging the some other population within the tissue specific one. So we cultured the MSC cells along with the we observed the epithelial cells also. Then again we can confirm this one. In conclusion of this one, the composite in subcutaneous is not vascularity is not developed, and the composite with the peritoneum is vascularity with the epithelial cell. Then we confirmed these things, or else peritoneum SS improvisation material in biocompatibility for esophageal regeneration. Material is suitable for esophageal regeneration. Confirm it again in the higher animals. And we developed a composite. That composite will help us to develop vascularity and tissue specific encouraging. Then we conducted an experiment with the help of Dr. G.V. Rao sir and Rohit Dama and Dr. Shashikala madam and all the people, and Dr. Ranjit also helping a lot on these experiments. So in details of the experiment, each and everything Dr. Rohit Dama will tell you better than me. And after resected of this one, wrapping the SAS material to the habit mode, then we observed that here are the microscope vascularity is developed here. So, and again the vascularity is confirmed by histological observation. It means that in vivo in the tissue specific conditions also the material is biocompatible and it's developed vascularity. And here we observed that some of the epithelial cells also. It's encouraging some other populated cells. Then again we completed this experiment, and again there is so many success along with the some constraints found that. Because the main thing is the material is biocompatible and vascularity is developed and intact cell viability and epithelial population also developed within the tissue specific region. Along with that, the constraints of this one, limited mechanical strength, because it's a single layer or double layer system, but maintaining the mechanical strength is very it's very difficulty. And incomplete regeneration. So we are not getting the complete layers of the esophagus. For example, we are addressing smooth muscle and striated muscle cells, very difficult. And risks of fibrosis, obviously that incomplete regeneration will happen to the fibrosis. And functional integration also very difficulties and long-term studies. So how to overcome these limitations again is a big challenge for us. To overcome these challenges, this time we tried getting the resected esophagus from our OT department, and then we can separate the different type of tissue layers from the esophagus, sided muscle tissue and the epithelial layers. Once we dissect the tissues, you know that it's very difficult to getting into muscle layer in the in vitro condition, especially in the esophagus. But in our department and our HF AG achieve this to developing the muscle layers, sided muscle and epithelial layers we develop here. Right now, we are focusing currently initiating the 3D or three-layer system for esophageal regeneration. At the same time, now we are addressing the three ways of the success of the esophageal regeneration. Once we wanted to develop taking the helping of a bioprinting technology, and another one is the decellularization recellularization technology to esophageal regeneration, and the using layer by layer technology that we are in the stages right now. So indigenously, and earlier I showed that we brought from the SAS material from the Cook Biotech. So source in GMP facility, we developed this SAS material indigenously. And again, this is a qualitatively packed EC. It means that this material is completely qualitatively. It means that this material is decellularized, and it is allowed to cell proliferation and biocompatible. Again, to addressing the parameters bioink and deceleration, recellularization technology purpose, decellular esophagus we prepared here, and the 3D layer of the different layers of esophagus is also prepared here, and we prepared bioink for preparation of 3D printing technologies. And finally, I concluded that we are enhancing biocompatibility for SAS material for esophageal tissue regenerations, and we developed a composite in vitro cellular biocompatibility was developed, and established a esophageal tissue specific cultures, and developed a decellularized esophageal ECMs, and integration of decellularized ECM with carrier material. And future studies are recommended. We wanted to go ahead with the 3-layer culture system by taking off what we said, the recellularization and decellularization, 3-layer culture technologies, and 3D printing technologies. I sincerely acknowledge to the Dr. Nagesh Reddy in AG Hospitals, Dr. Jeevarasu, especially in the AG Hospitals Director, and Dr. Shashikala Madam, and the faculty, doctors, staffs, and especially my team also. Thank you very much.
Very good afternoon. We did a small experiment with few, I think 4-5 cases, where we had rabbit as the animal. What composite layer they were speaking, that was basically the esophagus muscle was removed, the mucosal tube kept inside, and this composite layer was then wrapped for about 2 centimeter of the rabbit esophagus. So this is how we started, and rabbits were given intramuscular anesthesia, and their vitals, pulse, and oxygen were monitored. neck incision, which is the rabbit's neck. A very thin figures for the muscles, and it was very difficult actually in first cases to identify the esophagus because it's such a thin structure, and it was very confusing with the other vessels around. And what we did, we passed the infant feeding tube through the mouth of the rabbit, and that was passed deep into the esophagus, and that way it helped us to locate the esophagus. You can see this is the that is in this numbers. All were longitudinal vascular structures, and once we identified that, idea was to get the whole muscle layer out, and the challenge was in that thin esophagus to keep the mucosa intact. The tube, and we're trying to get the muscle fine dissection, fine in the infant feeding. You can see all around circumferentially, full muscle was removed for about 2 centimeter. With the artery which has gone around the esophagus, which is looped and still getting the air out from one side. Find insulin syringe section. Finally, we could get all around. One patient we had mucosal injury which was bad immediately. Muscle and just centimeter, it is only a mucosal tube. Mucosal tube. That was suture for. This is the area where this is the composite given about its described. These were then and nicely wrapped all around the esophagus for posterior to the esophagus. A flimsy layer that had to be nicely wrapped around without tearing it, and we used 2 sheets wrapped all around the this. And doubly wrapped, and another stitch was taken just to anchor so that it does not slip vertically up or below. Picture after it is one stitch we took just to fix the composite around that area. Then just push this back. I think about 2 weeks later we had sacrificed these animals, and Dr. Shashikala was telling it grows vessels and epithelium. So the future plan is we can get the muscle also. Be like getting a good covering for that tube, and that is what we are planning.
Thank all the speakers, and then I think we had an interesting session. Any questions?
Next week we'll be having same 3D bioprinting on clinical applications. The focus there will be liver.